at East Genomics, we're doing a set of events to help people better understand genomics and start to better be able to help each other provide genetic diagnoses for our patients with rare disease, but also our patients with cancers as well. So I'm going to share my screen. I've prepared a few slides, but I didn't introduce myself, did I? Sorry. So my name's Kate. So I'm the lead clinical scientist for the East Genomic Laboratory Hub. And so this is the region of England that you're sitting in, I guess, and we provide genomic testing for you. So the first thing I guess I wanted to do was to just ask some questions to you. So this is Genomics Conversation Week, and so we're supposed to be having a bit of a conversation. So rather than me just sit here and talk, I thought we could ask you some questions. So we've got a poll. And the first question that I wanted to ask you is if you'd had any training in genomics at all. So I'm guessing you're all healthcare providers, healthcare professionals across the NHS. So have you had any training? I hope you can all see that poll. Shall we try and get the results in? Can we do that, Laura?
Is that working? The responses are in the chat, I think.
Oh, let's have a look. Sorry, we didn't practice in this, anybody?
We didn't, sorry.
So, so I can't see the results. What are the results, Laura?
So we've got 56% have said yes and 44% have said no.
Wow, okay, so we got about half and half today. That's great. So Laura, you're going to have to help me and be my assistant for the next bit as well, please. So the next question I put up is rare diseases affect how many people in the population? So 1 in 1,700, 1 in 170 people, or 1 in 17 people in the population? I feel like I should play some game show music.
Okay, we've had 10 responses. We've got 70% who have said 1 in 1,700 people, 30% have said 1 in 170, and nobody has said 1 in 17.
Wow, okay, so maybe my question wasn't phrased well. So the answer is rare diseases affect 1 in 17 people in the population. So what I meant to ask was, and I think that it was probably misunderstood. What I meant to ask was how many people in the population are affected by a rare disease. And although individual rare diseases are really rare, that's why they're called rare diseases, actually they're really common in the population. And we'll have a look at some of those statistics in a minute. So can we go on to the next question, Laura?
So the next question was, our DNA sequence is represented by 4 letters, and these are ATCZ, ATCG, or ATCQ. So we've got 92% have said ATCG and 8% have said ATCQ.
Brilliant. Okay, so we're all remembering our school day ATCG genetics sequence, and we'll think about those sequences in a minute. Thank you very much, Laura, for helping me with that. That's great. So if I move on, we're gonna look at some of these statistics and that we've just talked about. So in the UK, we define a rare disease as one that affects less than 1 in 2,000 people in the population. But collectively, rare diseases actually are common, and they affect 1 in 17. We think that's the number, 1 in 17% of the population at some point in their life. So we all likely know several people who have a rare disease, a rare condition. We take those numbers, that means that there's 3 million people in the UK who are affected by a rare disease. So this is a real, a lot of people having a big impact on our patients, our families of those patients, and our healthcare system. And 50% of those patients with a newly diagnosed case of rare diseases are children. So we're often finding rare diseases in children. And we know, and as we said, that rare diseases themselves are rare, but we think there's about 5,000 to 8,000 different rare diseases. We're finding more and more as we move forward, and some people's rare diseases are unique to them. And one of the reasons why this is Genomics Conversation Week and we're talking about rare diseases is that 80% of rare diseases are known to have a genetic origin. So this is where rare diseases and genomics come together. So one of the things that I wanted to talk about today was, what does a genetic diagnosis mean for a patient? And also, what does that mean for their family? So when our patient, our little blue person, receives a diagnosis, a genetic diagnosis for a disease or a disorder that they have, then that can affect the treatment that they might receive. It might affect the healthcare professionals that they are referred to look at certain symptoms or to make sure that they're not having problems with certain parts of their body. The genetic diagnosis might affect the prognosis of that individual in terms of the likelihood that that they're going to reach some milestones and how treatment can be affected and can affect that prognosis. So that's for that individual patient, but also for that family. Having a genetic diagnosis really, I mean, it's really key that it provides information. So sometimes a genetic diagnosis doesn't provide a treatment option, but having information can mean that that family can reach out to families who are have similar diseases and perhaps identify clinical trials that they'd like to participate in. And also a genetic diagnosis means that a family can start to think about making decisions about if they want to get pregnant again and think about family planning decisions that they might make. So that genetic diagnosis has impact for that individual patient, but also for that family. And then when we think a little bit wider, perhaps to the wider family, but also to the wider population, finding a genetic diagnosis in our initial patient might provide evidence to lots of different people in the population who have a similar genetic, a similar condition where the same genetic change is identified, and that can provide information to that family and that individual within those families. So having this genetic diagnosis can be really important for lots of different reasons in healthcare. So how do we find the cause of our rare disease? So we're going to think a little bit back to our genomic training here. So we as human beings have cells, and in each of those cells we have a nucleus which holds our genetic information. And our genetic information is held in things called chromosomes. And when you pull out the DNA strand from a chromosome, we get this DNA strand with our nucleotides, so the A, T, C's and G's. We've got a different order here, but A, T, C's and G's. And so this is the genetic information that we have in every cell of our body that makes us who we are. And genes within this that are encoded by the DNA strands are translated into proteins that make our cells, make our hair, make our different different parts of our bodies make our cells do different things depending on where they are in our bodies or different times of our development. So a genetic disease can be caused generally by a variation or a change that's happening in that DNA strand. So we know that duplications of whole chromosomes can cause rare disease conditions. And at the same time, a single nucleotide substitution, so an A turning into a G in a gene in a really key place, can cause somebody to have a rare genetic disease. So we call genetic changes variants. You might have heard people talk about spelling mistakes. You might have also heard people use the word mutation. And within our rare disease space, we try not to use that word mutation for genetic changes that cause rare diseases. We save that for our cancer colleagues who are looking at genetic changes that cause cancer. So this is a really good way to distinguish between the two. So we use the word variants or variation when we're thinking about rare disease. So depending on the genetic change and what that does and where that does that in the body can depend on what type of genetic disease you might have. So we just talked about our DNA strand in our cells. That DNA gets turned into something called RNA, which is a photocopy of that DNA. So each gene is photocopied. And then that photocopy goes to the production office and a protein is made out of that RNA. And so if a genetic change produces the shortened copy of that RNA, then perhaps a short version of the protein is made. Or if that genetic change in our DNA means that that RNA isn't actually photocopied and the photocopier stops working, then actually perhaps no protein is made for that gene. And of course, our body is made up of hundreds of different types of cells, and some cells, the reason why we've got lots of different types of cells is because different genes are switched on and off in our bodies. So depending on where the genes are switched on or off within our body can change whether or not, well, it can change a cell type to change a cell type, but it can change where we're affected by genetic variants in specific genes. So I've got two examples now of rare diseases that we're aware of and that we know about and that we're routinely testing. And I wanted to try and show you the different ends of the spectrum of the type of genetic variants that we identify and how we test for them. So the first I thought I'd show you was a child's, well, there's a little cartoon of a characteristic features of a child with Down syndrome. And children affected by Down syndrome have 3 copies of chromosome 21. And so they have a total extra copy, and you can see in this what we call a carrier type. If you go down to 21, you can see that there's 3 chromosome 21, whereas all the rest of the chromosomes, there's only 2 copies are represented. And for a child with Down syndrome, a diagnosis can be really important because whilst I think we're used to seeing children with typical facial features, perhaps a little bit of short stature and some intellectual disability, but also children with Down syndrome can be affected by cardiac conditions. They can have issues with hearing and vision. And so ensuring that a child is diagnosed with Down syndrome and then can seek help from those different departments within the hospital can be really important. So when we test for Down syndrome in a newborn child or prenatally, we can either do a carrier type where we look at all those chromosomes, or we can do a PCR test where we just look at specific parts of chromosome 21 and we say, do we have 3 copies of that part of chromosome 21 when we're only expecting 2 copies in an individual who doesn't have Down syndrome. So this is one example where a chromosome disorder, sorry, a chromosome duplication, sorry, an extra copy of a chromosome can cause a child to have clinical symptoms associated with Down syndrome. So the next example, I wanted to find a very specific letter difference in the DNA code that caused a disease. And the one I was thinking about when I put these slides together is achondroplasia. So we know that there's one G, and if that G gets turned into an A in this sequence that codes this FGFR3 gene, then that individual will have achondroplasia. So achondroplasia is dwarfism, so a short stature, generally shortened legs and arms, but a normal sized torso. And this is caused because this nucleotide changes the protein code, and it makes the FGFR3, which is the fibroblast growth factor receptor gene, it makes more of that protein and increases bone growth in the skeletal system of one of these patients who have this disease. So that's two examples of how we find the cause of a rare disease. But the other thing I wanted to say to you today, and this is why we're here, is actually we can't find the cause of rare diseases unless patients are recognised as potentially having a rare disease and are referred to us for testing. So I guess this is my picture of you all, healthcare professionals across the NHS. So one of the things that we'd like you to do is to think about a little bit more about rare genetic testing in rare disease, and think about how you could start to think about what patients you see that can be maybe eligible for testing. So in the NHS, we now have a genomic medicine service where genetic testing is centrally commissioned. We have a very high-tech test directory, which is an Excel sheet that lists 373 clinical indications. And there's a PDF document that goes with this, which lists the eligibility criteria of patients who are eligible for those genetic tests. So you can have a look at this and go and have you can search in Google genomic test directory, and you can find the test directory Excel sheet and the eligibility criteria. And you can have a look and see what kind of patients you're seeing. The test directory's got lots of columns, so you can split it into different specialisms. So like musculoskeletal, hearing, hematology. So there's different specialisms that are represented within the test directory. And so I guess what I wanted to finish on is that if you really want more information and want to start thinking about how genomics can affect your practice and your patients, then I think the great place to start is this genomics education program. And so I've put a QR code here and the website here. So that's it from me.